Well, good afternoon, everyone. Thanks for coming along. It's great to see a lot of people showing a strong interest in biostimulants, and that's something that we've seen over recent years. I'm Richard Salvage from Maxtim and a research student at Queen's University, and with me I've got Dr. Colin Fleming from the Agri-Food Bioscience Institute and Queen's University. So we're going to talk about biostimulants and how they can fit in and enhance existing protocols and regimes and treatments. Okay. So over recent years, we've seen a significant increase in the use of biostimulants, and that's been driven primarily by a real desire to reduce negative impacts on the environment. But also, as I'm sure you're all aware, there's a much more reduced availability of agrochemicals. And it begs the question, what else is out there and how can we maintain the standards that we've set ourselves? So that the agricultural industry have been using biostimulants for some time, and they've been used for a wide range of crops, increasing quality, yield, and plant health, and as well as quality. Now, all this knowledge is available to the turf and amenity sector now. Don't know if I can get it to work. Okay. It's a big market. By 2025, it's anticipated to be in excess of $4 billion, and a large portion of that is going to be used in Western Europe. So far, the important crops that biostimulants have been used on are combinable crops like cereals, high-value crops, which includes turf. So it's a big market, and that means there's a lot of big players who are interested in developing high-quality biostimulants for it, and they're available for the turf industry. Now, if we look at how the yield has progressed over time from plants, over the last 50 years or so, we've achieved what we've now got as a current yield by using pesticides, plant control products, and high-quality fertilizers. But we know that there's a genetic potential in just about every plant. And in order for us to achieve that, to achieve the optimum situation, we know that we need to overcome abiotic stress to do that. And this is precisely where the use of biostimulants comes in. So there's still a big gap. There's still a lot that we can achieve using natural products. Now, some of you have seen biostimulants develop over time. Some of the first ones were not really living up to expectations. And one of the big issues here is when you've got natural products, they can be difficult to manufacture consistently and repeatably to get that performance that you become to rely on. So it makes sense when you're selecting a biostimulant that you work with products that can demonstrate repeatable and consistent results. One way of doing that is to make sure that the supplier has got a very strong control of their supply chain to make sure the raw materials are consistent, and that it will help eliminate some of the issues of bad performance. So, what's happening on the regulatory front? Well, there isn't any at the moment, but it is coming. Later than planned, but it's definitely coming. And the likelihood is it's going to be part of the EU fertilizer regulations. And in order to meet those requirements, it's important, the biostimulants will have to demonstrate their efficacy and safety for crops and turf. So any major manufacturer will have to comply with the European legislation to gain access to the wider market. And this is irrespective of Brexit, because the UK is only a small part of the market. So anyone out there will want to comply with whatever the European standards are. So, as I said, at the moment, there is no standard regulation in Europe across the piece, but there are different approaches from different countries, and they will be unified at some point. The European fertilizer regulation only covers mineral fertilizers at the moment. The proposal is that there will be 36 new product function categories, PFCs, and the important one for us is the PF6, which covers plant biostimulants, and that will cover microbial, non-microbial, organic and inorganic materials. So when you're looking for a supplier to engage with, there's some key things that you need to look at. Try and ensure that you've got strong, repeatable data, that the mechanism of action of the biostimulant can be demonstrated year after year. I know we get some strange years, and last year has to be classed as one of them, with the beast from the east in the springtime and then a massive drought in the summer, but you're still looking for some kind of efficacy there. You also want to make sure that it positively impacts the phenotypes which are important to you, and that the organization is ready to comply with the upcoming regulations. Bit of a dodgy switch on this, isn't it? Okay, so when we look at the biostimulants and what do they act on, which are the phenotypes that are important to us? Speed and homogeneity of germination, growth of the plant, chlorophyll production is key, leaf length and plant size, and most important for me is the root development, because essentially if you can get that root system working well and big enough, it'll support a lot of stress and support the plant growth. But you should also expect to see some better tolerance to a range of stresses as well, both biotic and abiotic. So drought, heat, salinity, and these are all key elements that we've seen in the last 12 months. And also fungal disease and pests. Now, the biostimulants won't have a direct impact on those, but it should help the plant health and help resist. Okay, so what I'm going to show you here is how that manifests in some mechanisms. I've got a short video of different formulations of biostimulants against control. So on the left-hand side of the pots, you'll see the control and various formulations. These were planted in the winter, so the temperatures were at 5 or less. So it took a little while to get going. So you can see the control here on the left, and we ran this until they germinated to validate the experiment. So what we're seeing is faster germination, more homogeneous germination, strong growth, and you can see a variety of color there indicating chlorophyll production increases. So we've talked a bit about biostimulants, what they can do, and what is key in some of the phenotypes. But how does that correlate to the plant physiology and biochemistry? And Dr. Colin Fleming is going to talk to you a bit about what happens inside the plant when stimulated by biostimulants.
Thanks, Richard, and hello, everybody. So what I want to cover really is how biostimulants actually work in plants, because to understand that, allows you to select the correct biostimulant and to use it at the appropriate time. So our research program at the university really has been targeted at identifying modes of action. As Richard said, in the past, the knowledge of how these things work has been very patchy, which in turn has led to patchy success in their use. And what we're hoping is that a more scientific knowledge will allow us to be much more efficacious in the way we use these biostimulants and give you guys much better results. Now, the first thing I want to talk about is a thing called plant priming. And plant priming is really plant memory. When I say plant memory, people think plants can't have a memory, but they can. And anybody that has actually used the hardening process, taking plants out of the glass house into the wider environment, is actually using plant priming or plant memory to ensure their plants are growing. And basically, priming or hardening or conditioning is it's a physiological state by which the plant is being prepared to respond to a future stress. So you're basically training the plant, or as another way of putting it, you're actually giving the plant a vaccination to cope with a stress later on in the future. And this priming or this hardening can be produced by a natural stress such as drought or heat shock or cold. You can use chemicals, and that's where the biostimulants come in. Or you can use things like transgenic mutation, but that's really out of the remit of this particular talk. And priming really is a thing called somatic memory. So plants do have a memory, and priming is the basis of this. So how does priming actually work? The signaling involved in priming is actually fairly basic for all plant types. So whereas a lot of the work has been carried out on agricultural crops, it's exactly the same mechanisms that are being used in grasses, which you guys are involved in. And the initial stimulus, for example, in this case, it's on a leaf, is caused by a stress, such as, say, cold or heat, or a priming agent, such as a biostimulant. That induces a thing called oxidative burst, which is a reaction inside the cell, which in turn induces a plant hormonal response. And this response can be spread from one part of the plant where the initial stimulus is to other parts of the plant. And what you see then is changes in gene expression. So the genes inside the plant that are allowing the plant to actually function are changing in response to this initial stimulus. And this really is the basis of plant memory. Now, this little diagram shows you how plant memory and this sort of initial stimulus works. So, for example, if you look at the red line down at the bottom here, this is a normal plant which has not been subjected to a biostimulant. What happens is, here's our response, our defense response, and the defense response can be against an abiotic stress, such as, say, heat, or it can be a response against fungi. The biochemical mechanisms in many cases are exactly the same. So here's our plant, and there's no defense reaction. The plant's happily growing away. You get a stress, so for example, a fungal attack, and what you find is the plant will slowly respond to that fungal attack up to a maximum response. So the plant defense system is being triggered here, and it's taking quite a while to build up. And by the time you've got the defense response really acting strongly, there can be a lot of damage to your turf. The rationale behind using a biostimulant is you give the plant a small attack early on, and after that initial priming, the plant defenses drop again to normal levels. However, when the stress kicks in, you can see you get an immediate and very strong defense response. So it's a bit like a vaccination. Here's your vaccination. Here's when you're hit by the disease, and you get a very strong defense response. And this is basically what you're trying to do when you're using biostimulants against biotic or abiotic stress. Now, I mentioned gene expression. The basis of all this is trying to encourage plants to change their gene expression. You want certain genes turned on and certain genes turned off. And this data is from some of the work we have carried out in the university. And you can see here, there are two types of chlorophyll in plants. This particular gene here, chlorophyll A, you can see here's our control. Add the biostimulant, and you can see there's an increased response. So there's more chlorophyll being produced. And this is a normal reaction to the use of biostimulants. You get increased levels of, for example, chlorophyll production. This gene here, chlorophyll B, you get an enhanced response. The response is much stronger. But both of these are where there is no stress. So the plant is growing happily, and you're getting some degree of effect when you use the biostimulant. Over here, however, we're measuring a thing called anthocyanin. Anybody who grows ryegrass will recognize the purple color in the grass, and that's quite often an indication of stress. Anthocyanin is produced as a response to stress. So quite often we will measure that purple color to tell us if our plants are under stress. And what we've got here is no stress, where here's our control. We add our biostimulant, and you can see the anthocyanin level has dropped slightly, which indicates we've reduced the stress in the plant. But the plant's growing happily, so the stress levels aren't that big, they're quite low. However, here we've got a stress, and this could be drought, it could be heat, it could be cold stress. And you can see that in the untreated plant, stress levels are very high, as indicated by a high level of anthocyanin. This time when we apply the biostimulant, we get a very big reduction in levels of anthocyanin. So where we've got our stress treated with biostimulant, we're getting a big reduction in stress. And that's what we find quite often. When you use biostimulants to protect your turf against a stress, you've number one got to pre-treat before the stress. Remember the inoculation analogy? So pre-treat, and there's got to be a stress to see the effect. So that's the two important factors. Pre-treat and only expect to see a result when you get a stress. If you don't get a stress, if your weather forecast has been wrong, you will not see any effect. So we've looked at how, in this case, primary stress signals, these are the biotic or abiotic factors, kick in. They cause changes in the plant cells, and these actually will cause changes in gene expression, and in some cases in the DNA itself. Now, these changes can be of different types. The first is called non-heritable, and that basically means is that if you treat a cell, those changes to the gene expression are confined to that cell, and that gives you short-term resistance against stress. So you've got your turf growing, you treat the leaves with your biostimulant, and anything that's been given the biostimulant, you will get short-term resistance. The other type of change is a thing called a heritable change, and in the medical field, this is called epigenetics. And in this case, it's different because this resistance response can be passed on. And again, there are two types. First of these is called mitotically heritable, and that means that the changes are passed on as new cells are being produced. So if you've got a root system that's been treated with the biostimulant, all of the cells in that root system are tolerant of attack. If you can induce this type of response, any new root that you put out will also have resistance to that stress. The third type of change is one which many people don't know about, and it's called mitotically and meiotically heritable. And what that means is that you get this resistance, tolerance passed on through the generations. So if the plant produces seed, the seed also contains that resistance. This is an amazing thing. It goes against all what we were taught in school as regards genetics. What it means is if you're growing annual meadowgrass greens, you've treated your turf, the potential is that the seed produced, which gives rise to the new batch of turf, will carry this tolerance. And this is a really major thing for future management of turf and other crops. So you're passing on an induced tolerance of disease or abiotic stress to the next generation of seed. And that's a really important tool for the future. If we look at a plant cell, this is basically a diagram of what goes on. So here's our stresses, biotic or abiotic, and they act on sensors on the cell wall. These sensors in turn, once they're stimulated, produce various chemicals in the plant or various signal transducers which affect the plant hormone system. So this is jasmonic acid, salicylic acid, abscisic acid. So they're making changes in the gene regulation and also gene changes inside the nucleus. So this is a diagrammatic interpretation of what goes on when you apply a biostimulant. So here's our abiotic stress, our biotic stress. They're affecting the receptors. And inside the plant, this is the type of reaction we're seeing. You're getting changes in the signaling pathway. So this could be jasmonic acid, abscisic acid. They're all being stimulated, either turned on or turned off. You also get crosstalk. So these pathways are not isolated. They're having an effect on one another. So you get some are increasing, and they will induce a decrease in another pathway. So you're getting all changes in these pathways. And also you get these interactions with proteins. And it's the interaction with the protein that are carried on to the next generation. And what our work really now is trying to find out what all of these changes are, how we can develop biostimulants to actually target these changes and give you guys really useful, fine-tuned tools to control stress, be it disease or heat, for example, or drought. So our program really is trying to understand how plant physiology is affected by biostimulants. We're trying to figure out the mode of action and identify the bioactive compounds. We take a seaweed extract that contains thousands of materials. Which of those are actually causing the effect you want? And that's what we're trying to solve. We're doing it by using a number of plants. This is one thing called Arabidopsis. It's a little brassica, and it's a genetic tool that we can use to determine modes of action. For example, here's a set of little plants that have been tested using or treated using biostimulants, increasing amounts of biostimulant. You can see when we put on drought, the no-treated plants are looking pretty poor, but as we use different levels of biostimulant, we're seeing enhanced tolerance to drought. And the same pathways in this little plant are being induced in grass. And we're measuring growth rates, we're measuring leaf size, root size, and this is the type of bioassay that we're using to actually determine mode of action. Here's one of our experiments. This is our control plant that has been given no water for a number of days. Here's our biostimulant treated plant. You can see immediately there's a big difference. A single spray of a biostimulant has protected this plant against drought. But the key thing is treating before the drought hits the plant. If you do it during the drought, no effect. So these die, these survive, and it's by the use of this priming genetic memory procedure has allowed us to achieve this. And I'll pass over to Richard for the final part of the presentation.
Thanks, Colin. So you can see that we're really keen to understand not only mechanisms of action, but also modes of action as well. it helps the design process for biostimulants. And what we've been doing recently is looking at the effect of adding biostimulants to other components and compounds to see what kind of synergistic responses we can get, if any. So I've got some examples of that to show you. The first one is a product we call a mycore system, where we've combined biostimulant formulation, liquid biostimulant, with mycorrhizae, fungi, and growth-promoting bacteria. Now, the first example I'm going to show you is on blueberries, and I know you don't grow those on your golf courses and football pitches, but as Colin said, if we can understand the mechanism in one plant, we can understand it in others. And I'll come on to talk about how they affect ryegrass as well. So what we have here is an untreated plant on the left, and this is a typical photograph from hundreds of samples. It's a blueberry plant. It was a plug. This was grown over 14 weeks, and with a combination of liquid biostimulant, growth-promoting bacteria, and it's specifically chosen mycorrhizae, we've had a significant increase in root development. When we split that down into each component part, you can see on the left-hand side the control. The next application was a single application of biostimulant, and we saw a step up. Then we added the ericoid root mycorrhizae and the biostimulant, again a step up. This is all evidence of synergistic activity. Then we added into the process the growth-promoting bacteria, and then finally the last set of results was based on all three, with then 2 extra treatments of biostimulant over several weeks. Ultimately, I think this demonstrates a synergistic response, and we increased the root mass by in excess of 200%. Now, when you've got a root mass which is significantly better developed than the untreated, you get a significant difference above the ground as well. So what we have here is 2 photographs taken on the same day in the same row, and the ones on the left had no treatment, and the ones on the right had the full 3-component treatment. But it was a one-off treatment, and the plant was allowed to grow for 12 months, and that's the result we had. It's quite significant. So applying that to ryegrass, we treated a range of many samples of ryegrass plants. We separated them from the substrate and dyed them in red dye overnight to help visualize the root system. We were then able to use a computer program to analyze the results. Again, this is a typical response from hundreds of samples. Using the computer software, we were able to measure root length, diameter, number of tips, and forks. So we got a really good understanding of the root morphology. And as you can see, the treated plant on the right-hand side was significantly more developed. And again, when you're looking at ryegrass, whether it's for a golf green or a football pitch, the grass is cut on a regular basis, daily, sometimes twice a day. So the stress that you're imparting is massive. You're taking away the plant's ability to photosynthesize. But if it's got a root system like that, the chance of survival and growth and renewed health is strong. Another example of where biostimulants can impact current regimes is here. We've developed a product called Trifix, which is an answer to reduced availability of pesticides. We've still got a problem with leather jackets and chafer grubs. So we have a bioactive, a wetting agent to increase the efficacy of the active, and also a plant strengthening biostimulant to boost the plant health. As Colin said earlier, the mechanisms, the modes of action in the defense mechanisms are very, very similar for biotic and abiotic stress. So the bioactive here is the entomal pathogenic nematode specific for those pests. And we've achieved some very good results using them. Okay, so how does this manifest in practice? So I just want to talk you quickly through a case study we have here. We've been working with Le Golf National, big year for them last year, hosting the Ryder Cup. We've been working with them for three years or more. And when we first started out, we did a complete route survey of the golf course. We took 60 cores, 3 on each of 20, 2 practice greens, and we took cores front, middle and back of the greens, so we were able to replicate the same position after treatment. They were exposed to Maxtim 3 times over a 9-week period, and then when we went back and we did exactly the same analysis, another 60 cores, we were able to then compare, and we noticed an increase of 36% on average right across the whole course in root length. We saw an increase in chlorophyll production, and the overall plant health was much improved. So we photographed every single core, and this was a typical example. One of the practice greens was treated half with Maxtim and half without, and you can see the difference in chlorophyll production. It's quite obvious. And Alejandro is very happy with the product. Okay, so we've demonstrated that complex biostimulants can impact biochemical pathways in defense mechanisms. These pathways are very similar for biotic and abiotic stress. And complex biostimulants can not only influence growth, development, and quality, but they can also assist plant health. Now, I believe biostimulants have got a big part to play in the turf industry. Standalone formulations can have a big impact on important phenotypes. And at Maxtim, what we're trying to do is to use biostimulants as a platform to build new and exciting products for the future. Thank you very much. Now, Colin and I are more than happy to take some questions. So if you want to turn his mic back on. Anything, or is all perfectly understood?
Would you recommend application rate
For what? Golf greens. It varies through the year. So a lot of biostimulants won't be effective if temperatures drop below 5 degrees, because you need the plant to be active to be able to take the product up. And also, the plant is much more active at different parts of the year. So what I'm sure you all know is you get a very active springtime. When summertime comes, and last year was a classic example, you get heat stress and no growth. So my recommendation is once a month in the dormant periods and once a fortnight in growth periods. So then you've got the priming all the way through the year. Anything else?
See a lot of your grass tests were on ryegrass, but will be maintaining low plant fescue for a lot of golf course breeds. So, were there any studies done on that, or you suggesting that it doesn't really matter what species, it should have the same effect regardless?
Okay, the question is about which grass varieties we've been testing on and are biostimulants applicable to those. And the answer is yes, we see a uniform response, and we have tested against other varieties. I've just shown you a very, very small part of our research here. Have you got anything to add to that?
I would say that it's clear that different species will have different responses to the same concentration of biostimulant. And one of the things we're trying to find out by understanding the mechanism will be to figure out what the appropriate dosage and application schedule is for different species. So we've looked at creeping bent grass, we've looked at Poa annua, fescues even, and although the general response is the same, there do appear to be differences. We're still trying to figure out exactly what the optimal is for each of those. There may also be varietal differences. That's something we haven't really investigated too heavily, but it wouldn't surprise me if there are differences. We certainly see that in agriculture, and it's an added complication, but it does highlight the need for good research to give you guys the appropriate tool and the appropriate control for these biotic and abiotic stresses.
Okay, well, thanks very much. It was a great session, and enjoy the rest of the show. And if you want more detail, we're in the blue zone. Maximum in the blue zone. All right, thank you.